Good morning. Today I'll be focusing on the human microbiome, but specifically the urinary microbiome and how it can potentially aid in the early detection of bladder cancer. Bladder cancer is one of the most frequently diagnosed malignant diseases, causing more than 160,000 deaths per year. Commonly affecting males 65 years and older, which is believed to be caused by an increase in life expectancy due to the advances in medicine. The scan on the right-hand side is showing a tumor in the bladder, which if you look at where the arrow is pointing, you can see a decent sized mass. The role of microbes within the human body is to maintain the overall health of the body and prevent the development of diseases. But changes in these microbes can also cause disease. These changes can occur in both quality and quantity. Changes in the microbes have also been linked to a vast array of diseases, such as diabetes, inflammatory bowel disease, cancer, and autism. The image on the top right hand side is simply demonstrating what inflammatory bowel syndrome looks like, while the image at the bottom right hand side portrays the effects scoliosis can have on the spinal column and the brain. Like many microbiomes, microbes within the urinary microbiome are abundant and can be difficult to characterize. With that being said, the urinary microbes typically associated with female microbiomes are more easily characterized than males. Typically, microbiomes that fall into the phyla such as firmicutes, actinobacteria, and proteinobacteria are the most abundant in the urinary microbiome, while genus such as Streptococcus and genus 1 through 68 are the most frequently identified. The graphs on the left-hand side are comparing microbes collected from healthy individuals to that of individuals with bladder cancer. As you can see, these samples typically share the same microbes, but the abundance of each microbe varies between the groups. The goal of the peer-reviewed article I picked was to characterize the urinary microbiomes of two groups, healthy versus bladder cancer. The researchers started with their hypothesis that the urinary microbiome can influence and detect the development of cancer. They gathered 36 urinary samples, but out of that, only 13 of those samples were not viable, leaving them with only 23 viable samples. 12 of those belonging to cancer patients or the experimental group, and 11 belonging to healthy individuals or the control group. After analyzing the samples all the way to its raw sequences, 252 base pairs were found. This resulted in 10 phyla, 19 classes, 26 orders, 61 families, and 107 genera being identified. When comparing cultures, a slight difference in the quantity of OTUs were observed, but there was a significant difference in the abundance of the OTUs. The control group was enriched with OTUs such as Streptococcus cristiatus, while the experimental group was enriched with Fusobacterium and Lactobacillum. Some were even completely dominated by Enterobacterium ca. This graph is basically showing what I previously stated, the changes in the elevation of certain OTUs compared to the control group and the experimental group. With the knowledge gained from the urinary samples, the researchers then tested tumor tissue, and 11 of those samples tested positive for Fusobacterium. Since Fusobacteria is known to promote an pro-inflammatory environment, it makes sense that those areas are prone to tumors. Fusobacterium is also known to prevent NK cell cytotoxicity and T cell activities, which ultimately promote immune invasions. Although there were no major differences in the microbiome's profile, this experiment did aid in identifying that the expression of some OTUs in the urinary microbiome can be indications of bladder cancer. This study proved that it is not the quantity of the microbiome, but the abundance. Some cons of this experiment were that all participants in this observation were male participants. There were no females, which means it's not a good indication of female microbiomes. And this study needs further studies and experiments to support its findings. And that's my references. Thank you for listening.